Assalamualaikum and welcome everyone to our presentation. We are from Group 6, Sen 1B, represent to you about one Nobel Prize winner's contribution in genetic and molecular biology for genetics and biology molecule subject, which is the code ENT1253. Our selected notable, which is Barbara Mcclintock, who won for Nobel Prize in Physiology or Medicine in 1983. These are our group members who has been led by our leader group named HAQQULYAQIN bin SAIFI. In our group, there are 9 members of ours including our leader. We will briefing to you about her biodata first. The real name of Barbara Mcclintock is Eleanor Mcclintock. For you guys' information, this winner is one of the successful women who get the Nobel Prize in Genetic and Biological Research. She was born in June 16, 1902 at Hartford, United States America, and she died at September 2, 1992 at Huntington in United States America. Barbara Mcclintock are also known as a success researcher and scientist in genetic structure of maize and corn. So, main focus of her successful is cytogenetics, which is breeding a maize using a jumping genes technique.
History of winning geneticists. First, Barbara Mcclintock is one of the members of the National Academy of Science in 1994. She was selected in 1994 and became the third woman to achieve this honor. Second, National Medal of Science 1970. This prestigious award was given to her for her exceptional work in biological science. Third, Thomas Hunt Morgan Medal in 1981. Barbara Mcclintock was awarded the Thomas Hunt Morgan Medal in 1981 by the Genetic Society of America. Fourth, Louisa Gross Horwitz Prize in 1982. This prestigious prize was given by the Columbia University for Barbara Mcclintock research. Next, Nobel Prize in Physiology or Medicine in 1983. Her discovery of mobile genetic elements and often referred to as jumping genes.
Next, History of Winning Genetics is Lasker Award for Basic Medical Research, 1981. The Lasker Award recognizes brilliant concepts and pioneering studies made 40 years prior which had monumental implications. Besides that, Kyoto Prize in Basic Science in 1985. Now for a discovery of jumping genus, this award one of the highest honors for individuals contribute. Thirdly, Genetics Society of America Medal, 1981. Lastly, Albert Lasker Award for Special Achievement in Medical Science, 1992.
Next is application of experiment. First is medical genetics. Understanding responsible element aids in studying genetic disease and creating gene therapy methods. Second is agriculture biotechnology. Maize genetic help in the development by modifying crops with specific traits as such pests or heat resistance.
The advantage of experiment by Barbara Mcclintock. The first one is understanding genetic variation. Barbara Mcclintock discovered mobile genetic element in plant more than 30 years ago. The discovery was made at the time when the genetic code and the structure of DNA double helix were not yet known. This is only during the last 10 years that the biological and medical significance of mobile genetic element has become apparent. The second one is human health disease. Mobile genetic elements have also been found in bacteriophages, viruses that infect bacteria, internoplasm, a type of parasite that causes African septic sickness, in insect, Drosophila. Several mobile genetic elements have been identified and shown to be closely related to genes found in RNA tumor viruses. The last one is investment in genetic research.
Disadvantage of experiments. First, delay recognition. Mcclintock's groundbreaking work on transposable elements was initially met with skepticism and lack of understanding from the scientific community. Second, impact on research funding. The skepticism and lack of immediate acceptance of her work had practical consequences, including difficulties in securing research funding. The last one is historical overshadowing. This historical overshadowing means that even with a Nobel Prize, her work is not always given the prominence it deserves in broader discussions of scientific history.
This picture is about a maze breeding that made by Barbara Mcclintock.
Case study of breeding on concordance. First, through observing and experimenting with genes in the color of concordance, she discovered that genetic information is not fixed. I sorted two genes that call as control element by tracking pigment change in corn and examining the plain large chromosome using a microscope. Second, she discovered that regulator elements can move to different positions along chromosomes and that these genes affect the behavior of neighboring genes. These transposable elements were responsible for new mutation in pigmentation and other traits. Third, provide the first experimental proof that genes were physically on chromosome by describing the crossing over phenomenon and genetic recombination. ZAMANI particularly those plants that produce variable colorness, perfect organism for jumping genes.
Discovered during the middle part of the 20th century by American scientist Barbara Mcclintock. Mcclintock on innovate cytogenetic technique allowed her to confirm Morgan's idea. Mcclintock studied the so-called ACDS system in maize. She discovered this system by conducting standard genetic breeding experiment using abnormal phenotype. Discovered was a chromosome break site, aptly named dissociation (DS). The move was controlled by the autonomous element called an AC. Mcclintock also observed many canals with colorless background amount of dark brown spot or stripe.
Next, these are the articles of our research about Barbara Mcclintock in discovering of jumping genes technique that she used in breeding a maize. In figure 2a, this article is about how Barbara Mcclintock changed controlling elements through transpose in genetic transposition of maize. To her, transposition was never in doubt, but others genetics quickly confirmed transposition in maize, and at the same time, a few of others considered it Mcclintock's major claim. Furthermore, in figure 2b, this article is about the importance of transposable elements to nurture original and unconventional thinking in science. Last but not least, in figure 2c, is a chronological list of Barbara Mcclintock that got major awards along become a student until she being a professional scientist in genetic and biological researcher. These are the references and research about Mcclintock that we use as a reference to finish our assignment. Hope that what we list here will help the viewers out there to add more information about Barbara Mcclintock. That's all from us, Group 6. Hope you guys enjoy watching it. Thank you for spending your time with us. See you again, everyone!